Today we dive into the fascinating world of embryonic development, specifically focusing on days 11 to 13. Let's start with day 11. Imagine a tiny entity known as the blastocyst. By the 11th to 12th day of development, this blastocyst is completely embedded in the stroma of the endometrium, the lining of the uterus. This is no small feat, as the blastocyst now produces a slight protrusion into the uterus's lumen, a true testament to the marvels of nature. Now let's talk about the trophoblast. This is a layer of tissue on the outside of the blastocyst, and it is characterized by lacunar spaces, or small cavities, in the syncytium. These spaces form an intricate network of sinusoids, and maternal blood begins to flow through this trophoblastic system. This is the establishment of the uteroplacental circulation, the life-giving connection between mother and developing embryo. In the meantime, a new population of cells appears, forming the extraembryonic mesoderm. These cells derived from the yolk sac cells form a fine, loose, connective tissue that fills all of the space between the trophoblast externally and the amnion and exocoelomic membrane internally. This sets the stage for the creation of the extraembryonic cavity, or the chorionic cavity, a key player in the following days of development. On the 12th day, the embryonic stage sees some dynamic changes with the formation of the chorionic cavity. This process begins with the development of large cavities within the extraembryonic mesoderm, a loose connective tissue derived from yolk sac cells. These cavities gradually merge to form the chorionic cavity, a new space that encapsulates the primitive yolk sac and the amniotic cavity. Now let's delve a little deeper into the components of the extraembryonic mesoderm. It's divided into two types: the extraembryonic somatic mesoderm, which lines the cytotrophoblast and amnion, and the extraembryonic splanchnic mesoderm, which covers the yolk sac. These linings play a crucial role in the formation and function of the chorionic cavity. Meanwhile, the growth of the bilaminar disc, a structure that will later contribute to the formation of the embryo, progresses at a relatively slow pace. It remains diminutive, measuring just 0.1 to 0.2 millimeters. Simultaneously, the endometrium undergoes changes known as the decidua reaction. The cells of the endometrium become polyhedral, packed with glycogen and lipids, and the tissue becomes edematous, or swollen with fluid. These modifications initially occur around the implantation site, but soon extend throughout the endometrium. With these changes, the stage is now set for the next crucial day of development. As we move into the 13th day, the surface defect in the endometrium usually heals, marking a key milestone in the development. However, there can be instances of bleeding at the implantation site. This is due to the increased blood flow into the lacunar spaces of the trophoblast, an outer layer of cells that provides nutrients to the embryo and develops into a large part of the placenta. Simultaneously, the cells of the cytotrophoblast, which is the inner layer of the trophoblast, begin to proliferate locally. They penetrate into the syncytiotrophoblast, the outer layer, forming cellular columns that are surrounded by syncytium. These cellular columns, with their syncytial covering, are known as primary villi. In the meantime, the hypoblast, the innermost layer of cells, produces additional cells. These cells migrate along the inside of the exocoelomic membrane. As these cells proliferate, they gradually form a new cavity within the exocoelomic cavity. This new cavity is known as the secondary yolk sac, or definitive yolk sac. It's much smaller than the original exocoelomic cavity. During its formation, portions of the exocoelomic cavity are pinched off, leading to the creation of exocoelomic cysts, often found in the extraembryonic mesoderm. Meanwhile, the extraembryonic coelom, the body cavity in embryos, expands and forms a large cavity, known as the chorionic cavity. The extraembryonic mesoderm lining the inside of the cytotrophoblast is then known as the chorionic plate. The only place where extraembryonic mesoderm traverses the chorionic cavity is in the connecting stalk, which is a precursor to the umbilical cord. By the end of the 13th day, the embryo has gone through monumental developments, setting the foundation for the complex life form it is destined to become. Understanding these early stages of development helps us appreciate the intricate process that brings life into existence.